Watch this video to know all about how antibodies are produced. It starts with a single cell extracted from a mouse. The mouse has been genetically modified, so it produces the antibody. The extracted cell would die in short time by itself as any other living cell. But there are cells like cancer cells, myelomas, that do not die rapidly. By fusing the mouse cell with these cancer cells, we create an immortalized cell line that produces our antibody. So we combine the ability to produce antibodies of the spleen cell with the immortality of the cancer cell. And this can be done in large scale in a lab and in big production batches. But before we go into detail, please make sure to like this video. It's for free and pushes it. Also, make sure to subscribe so we can make more videos like this. Thanks. When an unfamiliar threat invades the human body, our natural defenses launch an immediate counterattack. The immune system instantly generates a massive wave of polyclonal antibodies. They deploy rapidly, but they form a chaotic, uncoordinated mix. The downside to this natural response is the lack of targeting precision. You get a wide variety of antibodies with messy, unpredictable capabilities. The ultimate goal of medicine is a treatment that locks onto one specific pathogen. We want to avoid random attacks and leave healthy tissue completely untouched. This is exactly where monoclonal antibodies change the game. They offer a precision solution designed to isolate a single highly effective antibody and clone it outside the body. These engineered treatments can lock onto complex structures, like cancer cells, with absolute accuracy. The process begins when an animal's immune system reacts to an introduced antigen, generating specialized B cells to combat that threat. Scientists then extract the spleen to isolate these active B cells. While these cells hold the exact defense mechanism required, they cannot survive long-term, meaning they possess zero natural permanence. The modern answer to this targeting issue is a monoclonal antibody. Instead of generating a massive variety of defenders, monoclonal antibodies are exact biological replicas, all created from the same original cell line. The specialized immune cells capable of binding targets effectively, known as B cells, have a finite lifespan. When isolated to grow medical treatments in a laboratory, they perish long before they can provide meaningful volumes of antibodies. So we use immortalized myeloma cell lines that possess the unique ability to duplicate and replicate forever. By combining short-lived B cells and immortal myeloma cells in a polyethylene glycol mixture, researchers induce cell fusion. The added compounds compel their cellular walls to blend together. The process must confirm monoclonality. High-precision initial screening filters out background noise, visually proving that a colony developed from a solitary starting cell. Once isolated, the line undergoes precise functional evaluation. Automated assays demonstrate that these uniform antibodies bind perfectly to the targeted viral or bacterial antigen. This substance forces the outer walls of the two separate cells to fuse cleanly into a single entity. This creates a brand new supercell, retaining the precise disease-targeting capabilities of the B cell, while gaining the endless replication power of the myeloma cell. This creates a unique cellular pairing that perfectly integrates the best characteristics of both lines. It preserves the exact antibody structure while ensuring endless replication. Automated analysis measures how effectively each line attaches to its specific target. This visual breakdown instantly isolates the top performers. Then, high-throughput robotic picking systems leverage initial imagery to locate that specific placement and harvest the highest-yielding strain. This uncovers the individual ideal clone that manufactures the required antibody. Automated software then ranks the validated selections by their total concentration output to determine the single most productive cellular line. The process must confirm monoclonality. High-precision initial screening filters out background noise, visually proving that a colony developed from a solitary starting cell. Once isolated, the line undergoes precise functional evaluation. Automated assays demonstrate that these uniform antibodies bind perfectly to the targeted viral or bacterial antigen, ensuring that the harvested line originates from a single, completely pure parental source. Next comes the scale-up phase. The individual line is moved into automated growth platforms and massive industrial bioreactors, where it quickly scales up into millions of identical copies. Cultivated within massive manufacturing vessels, that single validated cellular factory divides indefinitely. manufacturing huge volumes of the exact same antibody. Large quantities can be produced in huge bioreactors. The process lasts several days and is monitored cautiously at all times. As antibodies are so specific, they go to their target in contrast to conventional medicine that can have a lot of side effects and destroy tissue around the targeted disease. Today, antibodies are used to treat certain kind of cancers, autoimmune sicknesses, and dangerous infections. But how do the antibodies reach the patient? This is done by aseptic processing. First, the liquid passes through an advanced filtration grid that blocks every spiked microbial contaminant, while allowing the vital antibodies to slip through completely unhurt. That is sterile purification. Simultaneously, their final destination is being prepared in an isolated unit. Intense thermal energy and chemical mist blast the clear glass containers, destroying any hidden microbes before the medicine arrives. So the target antibodies are clean and the containers are completely clean. But how can they combine safely when human staff constantly release invisible airborne particulates? They are brought into a strictly guarded, closed-system environment. Inside this sterile clean room, robotic equipment acts as automated protectors. They handle the fluid transfer tasks entirely on their own, completely removing dangerous human contact. Immediately following the fill cycle, the machines apply an airtight seal, locking the secure chambers down tightly against outside threats. And that collection of pure antibodies now sits completely safe within their protective glass home, totally sterile and ready for clinical use.